Yeah, hi, just really quickly, I'd like to bring up the second set of focus discussants, Rick Myers, Katie Pollard, and Danny Fallin, to talk about bridging day 1 and day 2 topics, so connecting discussions about variation, function, and phenotype.
Somebody pointed out that it's a little ironic that NHGRI put me in a group that has the word focus in it, but nevertheless, we'll try. This is a little bit different as well. The purpose of the session, from what we understand in talking to NHGRI, is an issue that they bring up in other contexts as well, is how can they de-silo their initiatives. They have lots of projects and initiatives, and while there is success in some of these talking well to each other, they're sort of administratively separate, and the idea is that we could get a lot more by putting them together. So that's what we're going to talk about, and we're going to talk just a little bit, and then hopefully have you weigh in on a lot of these ideas. So this is just a partial list of projects that are either funded by NHGRI alone, funded by NHGRI plus another, or even other ones not funded by NHGRI, but where they play a role. And there's a lot more of these. And you've seen these, and many of you are part of these. So we thought we'd start off with some successes, some places where there has been good integration, and this is just a couple of those that we thought of and talked about a little bit, and that's the model. What you heard from Hugo this morning, this really nice connections between the model organism programs and Mendelian centers, or at least a center, and UDN, and even talked about that across the country, not just at his institution. So clearly that has benefited those projects. There could probably be a lot more integration between them. And the same thing with the mouse knockout project and the Mendelian centers, and there are clearly others, so think about those when we're having our discussion in a couple of minutes. There are other ones with NHGR and other institutes, and so Tim Reddy's discussion, I asked him how this came about his NIDDK project, was really introduced by discussions with NHGRI first, and so things like that do happen. The question is, can there be more of those? The target 2 gene by environment and TopMed and many others are relationships between NHGRI and other institutes. And one of the points that we think is important that we want to talk about with the whole group is that while it happens this way sometimes, it doesn't always. NHGRI and these scientists and the ones funded by these groups often have ideas. They should pitch these early on so that the study designs can mesh with all the programs. Sometimes they happen after the fact, and there's sometimes problems because of that. So some of the places, and there are lots of these that you can list, and we want you to contribute to these, are where there are silos, which is a positive way of putting that as opportunities for improvement. So one of these is functional genomics, and all of the functional genomics type projects on meshing those better and more formally with the genotypes, genomes, and phenotypes, including clinical ones. The other was, and this is done a little bit, I know I'm part of Encode, so I know that we try really hard to do this, is to match cell lines or tissues or organisms and developmental stages across all types of assays. So that, and that's done some, but it probably could be done a lot more. And there are many more examples where we want audience participation. So Danny, I think you're going to come up and lead this part now. This is where we want you to give your ideas about places where you see problems and where it could be better. Oh, and I should have mentioned, this is not just about existing programs. This is thinking about the next 5 years and the next 10 years as well. So think of it that way, please.
Yeah, and just to get the conversation started, I wanted to emphasize silos exist for good reasons. They're not random. So I think this is hard. And I think, at this point in the meeting, it would be really great to talk about the hard stuff, because it's great to be sort of blue sky and upbeat, but I think there are some real barriers, some of which have been discussed and some of which haven't. So I'd love to unearth a little bit of that and think about what could be different in the next 10 years. And we're going to start with these programmatic silos. between like programs or research programs and research areas. And then we have a few other silos to talk about after we have some discussion on, maybe 20 minutes or so on this, some other types of silos as well. Thanks. So.
My job is not to talk, is to synthesize. So this is the time for you guys to start throwing out examples of where you think there are sort of the day one and day two of this meeting kind of thoughts that are already working together in a programmatic way, and places where you realize they aren't working at all yet programmatically together through the lens of NHGRI. Okay.
Yeah, I think even
Identify yourself, please.
Oh, Eric Jarvis, Rockefeller. I know that the mission of the talks or the discussions for day one and day two were meant to focus on different aspects, but I think they have sort of converged, hearing the same comments on yesterday and today. And if I had to speak up my comments, I'll probably say the same thing I said yesterday and today. And so what I'm wondering is that, I mean, that's not the case for everything that was discussed, but I would, should also recommend that maybe going beyond those initial comments and what you're asking for, what are the hardcore things, making even, let's say, recommended hardcore support for things, whether it's complete genomes or complete phenome and so forth to genome in this session. I'm not trying to get you to change it all, but it's just recognizing that there's been a lot of convergence in the conversation. And it'll be probably not good to hear the same thing over and over again.
So in
Identify.
Andy
We know
Clark,
who you are.
Cornell University. In the breakout session I was in yesterday, we talked about genotype by environment interaction. And of course, we did talk about NIEHS and their efforts in that area, with the mention that it seemed like there was more of a toxicological approach, that there's sort of various environmental toxins that we're concerned about. That was sort of their purview. And that there's a very ripe area for some kind of joint project between NHGRI and NIEHS, where we would bring to, the NHGRI group would bring to the table a much more rich molecular sense of the phenotyping, perhaps even more along the lines of modeling. That is a particularly ripe area because we're recognizing that G by E is something we haven't paid enough attention to. They've started to, but in a perspective that isn't as deep as we could go, and so it's just waiting to be done.
You're gonna call? Okay, go ahead. That's okay. Eris.
Hey. Yes, Eris Aiden, Baylor College of Medicine. I want to talk about what I think is a really major silo, which is the distinction between traditional sequencing-based omics and imaging and microscopy data. In the past decade and in past decades, it has certainly been true that the kinds of data that's been produced by microscopy in many ways was so different from omics data that they really could not easily be compared. I think that barrier is collapsing. I know even in our own work together with Ting Wu and Marty Renum, we were able to recapitulate high C results, but not using a sequencing-based approach, using a serial fish-based approach. Similar sorts of, many beautiful stories of this type exist, with RNA-seq and all sorts of other technologies. And I think that this is going to become much more the rule rather than the exception, especially insofar as microscopy-based methods almost automatically give you things like single cell, give you exact positioning, things that people really, really care about increasingly. So I think figuring out how to break down that silo is going to be very, very important.
So what's the, I agree, what is the barrier to a technological silo like that? Is it just history? These people haven't spoken together? Is there like sort of religiousness about things being better than other things? Is it an interoperability, a comparability issue? I'd like to hear some discussion from a variety of people. If we wanted to bring together genomics and imaging and make imaging more central part of NHGRI, what problems would we have to overcome? Yeah.
Yeah,
Stephen Chanik from the NCI. I mean, this is a long-standing issue in cancer where we have both imaging with respect to radiographic images and then also all the histopathologic H&E, the molecular analyses per se. And I think a lot of it has been cultural. But what's been most challenging, to be quite honest, is understanding what the questions are that are going to be addressed, because we have very large studies. Our group has just published 100,000 images of women who are HPV positive in their services, and it's 96% for the AUC for predicting who's going to go on and develop cervical cancer. So AI is very powerful, but the question is how you apply it and what specific question. And the hardest thing has been agreeing to what question is going to be addressed. And for that, the success has been in the long perspective studies where we have outcomes that we can work backwards from. If you're setting these up, it's a very difficult thing, and we can see in the NCI having a lot of difficulties trying to get these three groups together: the genomic characterization with, radiographic and digital imaging of pathology, to be able to agree what questions they're going to ask, whether it's classification, whether it's outcome, whether it's response to therapy. There are fundamentally different questions, and that's been a very difficult thing in a place where you'd think it was obvious and would work very easily.
And is that a cultural barrier, or we're too broad or technology-driven and not enough biology-driven problem?
I think it's a cultural more than it is a biology per se. What's the perspective of what the pathologists want to know versus what the radiologists want to know versus the clinical trialists versus the cancer biologists and or genomicists who are looking at this. They have very different questions that they're trying to ask. And so it's taken long conversations to get everybody together to agree what you can and can't do with the data sets that are in hand, much less plan what you do next, okay? I mean, there's a fair amount of discussion that has to take place. It just doesn't happen overnight.
Go ahead, Katie.
Please.
I want to echo that. It came up in our small group earlier and also in this larger group in the previous session that there's that kind of cultural difference even in the room. Some folks who are very uncomfortable with types of research that aren't really geared to particular mechanistic understanding versus others who think that there's utility in measurements that can do a particular thing even if you don't understand the connections between the two of them. And this gets to the prediction modeler kind of folks versus the etiology or the causal mechanism folks. I think it's all related in that cultural divide.
Yeah. I mean, we've had this in GWAS where those who have argued, do you have to understand every variant, or is the polygenic risk score far more useful than what we've learned from any individual variant that's been pursued? That continues to be a major discussion to this day.
I think just, do I push that? I think just, maybe this is working, yeah, that one comment, Erez, one example that you could do, you gave one example of the type of thing with imaging, and maybe not right here now, but things like this come up with multiple places where what are the questions that can be asked, just like he was just saying that because, and I think the cultural problem is it's our own making. You simply get people in the room at the same time and say what is possible, not what we have to do, but what's possible. And that ends up making people make those kind of connections. I think NHGRI can and needs to stimulate those kinds of meetings, and they're fairly simple, I think.
Yeah, just to riff on that, I think starting with biological motivation and designing to answer that question is fantastic. What I would like to hear is some thoughts on what could NHGRI do. Is it money, or just asking the question, or just requiring something? Is there something they can do to make people go a little bit further, answer their own question, but then go a little further to make their data also useful to a lot of other people? Like what that last little bit that would make it more useful to other people sometimes happens and sometimes doesn't. Is it just that it's too costly and it's not your priority, or it would really mean ruining the experiment from your point of view? What could NHGRI do to incentivize? You want to call on people?
Sean Burgess, Genome Institute. Carol Bolt mentioned this yesterday or earlier today. There's a fusion taking place across the model organism websites, the Alliance for Genomic Resources. And this is happening I think it's very good. I'm on the scientific board. I think there's a lot of great stuff that's coming out of that. It happened for two reasons. One is some people believed it was important, and two, Genome gave a combination of incentive in money and threat with guns to make it happen. And so if you really have a big initiative that you want to happen, I think Genome needs to step in with the money to help make that happen, the supplements, but also the threats that actually got the people in line to do those things. So I think it's both things are needed for really large, complicated problems.
Yeah, and threats can feel a little less aggressive if they're part of the announcement for the funding in the first place instead of like after the fact. So yeah. Oh, maybe someone who hasn't spoken yet. Is there anybody? Okay, Andy.
Well, yeah, just a quick point, particularly because we're talking about a 10-year time plan. The best place to put your money, if you're interested in solving the silo problem, is training. I mean, it's the young people.
That's our next slide. Okay, absolutely. Let's hold it for a second because I'd like to have a whole discussion about that, actually. Yeah. A couple other points before we go there.
In terms of silos,
Identify yourself, please.
Nancy Cox, Vanderbilt. In terms of silos, I mean, there is a field called genetic epidemiology, but let's say it's heavier on the genetic than the epidemiology in many current practices. And to get at more of the environment, we may be in some unique positions now as we talk about, collecting a lot of these data where we're going to have the genomic information, the opportunities to start distinguishing the sort of baseline part of this that is driven by the genetics from concepts related to exposure, I think is really, there are some great opportunities. And if we just start trying to work on including more data where certain kinds of exposures have been measured, I think there could be a real opportunity to create exposure type measurements in much more data than we've really collected it. Basically to invert the genetics, condition on the genetics and look at the rest of the space, as long as we know how to make some of the translations. Yeah. So some of the research on the methylation patterns observed in smokers is a great example of something that can be translated in lots of different ways with omics that could be collected. So I see that as a way to both break down silos and try to get more measures of environment and exposures that we don't make directly, but we learn indirectly how to make.
Go ahead, you call.
I can't see who that is. Yes.
Yeah, so this is Martin Hurst, University of British Columbia. I just wanted to make a pitch, and I know this has been discussed before, but I think one fundamental aspect of breaking down silos is interoperability, and that comes down to standards. Developing the standards, implementing the standards, and enforcing the standards to some degree, but also developing the tools for validating. I mean, I think anyone who's gone through the data submission process, knows how challenging it can be to actually get the metadata in a format that's acceptable. And then everybody's doing it in different ways. Even within the consortia that I work in, we develop the standards, and then people interpret them in different ways. And I think as a community, we need to get together. I think NHGRI could play a leadership role, maybe getting workshops together to help develop the tools to allow researchers to do this more effectively.
Okay, I think.
A little more.
Oh, okay. Maybe one or two. Okay, those two, and then we're going to move on to the next. Or was there
Marilyn
a third
Ritchie, University of Pennsylvania. One other place that I've heard mentioned at the meeting, electronic health records, and that there's a lot of need there for machine learning and natural language processing, but I think that's another silo where the medical and clinical informatics community is building a lot of tools for AI and machine learning and NLP in that space, and they struggle with knowing what questions would be appropriate to link with the biobank specimens that they have. And I think a few institutions are doing this well, certainly the ones that participate in eMERGE. But I think if we can partner more with the National Library of Medicine, who funds a lot of those clinical and medical informatics programs, that would open more opportunities. And maybe even in that training space, thinking about training people in both medical informatics and genetics or genetic epidemiology to build that bridge.
Great. So somebody was raising their hand over there, maybe not. Okay, okay, you and then John, and then we have to move to the next topic, please.
Marilyn Miller from the National Institute on Aging. So we're kind of where the other Marilyn is, looking at very large pieces of data, imaging, clinical, all kinds of phenotypic, biomarker, genetic, genomic, you name it, and many people doing experiments on these with all kinds of different ways of measuring the data, and they're being in multiple different databases. We're at the place now where we have so much data that we're going to have to move to machine learning in order to analyze the data, and our problem now is, because we didn't think ahead and hope HG can think ahead, that we need to, just as was said over on this side of the room, we need to begin to standardize, and you should standardize early. We didn't do that, and now we're going to have to go back and reinvest in that problem and get all of the data together, because we've been advised by our machine learning experts that we have to have the data accessible from one place. Otherwise, it isn't efficient, because silos are very costly for that kind of analysis.
Good point. A brief one, John.
Okay, this relates to silos and the environment by gene interactions that have been a topic yesterday and have come up again today. There are two huge studies: the health professionals follow-up study, 51,000 men, the nurses' health study, roughly 50,000 women. They have been followed for between 30 and almost 50 years, or 40-something years. Dietary questionnaires every year, continuous follow-up, repeat sampling, everything. All of the, in theory, all of the environmental measurements that one would like to make, that one could possibly reasonably make, have been made on these populations. And so getting something like that, understanding what is the barrier to bringing sequence information as a starter from those populations out into the world where people can operate on it. I think if you could solve that problem, you'd go a long way to addressing a lot of the things that have been raised.
That's a good example of taking things that already exist and putting them together and using the same kind of assays for everybody.
Yeah, does anyone in the room know the barrier there?
Well, at least several years ago, part of the barrier was nobody asked them.
This is Sharon Plon.
Sorry, Sharon Plon. Because I was discussing someone in their group that entertains the projects. They had gotten classic epidemiologists tend to approach them with projects. I can't think I've been now attending council meetings for almost 2 years. I can't think of a project that's been proposed that we ever discussed using the data. So part of it is for this community to be aware those data sets exist and to go to them with proposals. So that's part of the silo, is even having different communities of investigators really aware of these rich data sets.
Yeah, so knowledge. Just to continue on that thread, is there anyone in the room who is part of a project, an NHGRI funded project, I'll say, and learned during this meeting about another project or initiative that changes how you're thinking about your project? I'd like to hear a couple more examples of discoveries of those silos or opportunities like we just heard about from Sharon. Anyone else want to share one? Over there? Yeah, over there.
I just wanted to say that the NHS and PHS is very well known to the cancer community. We've scanned maybe 20,000 of their subjects in the 15 different cancer-specific GWAS that we've done. So, and they're being sequenced as well for certain select things on the cancer side. So they are open for business, and Walt Willett and Mayor Sanford are wonderful epidemiologists. Just make the representations to them, and they're very keen to look at diabetes, heart disease, you name it. So it is there. I think it's just the silo is a knowledge silo rather than, an inability to do something. Okay?
Dana?
Dana Crawford, Case Western Reserve University. So I would also posit that one of the silos is not just knowledge of these more, what I would call more traditional epidemiologic-based cohort studies, but also the way in which investigators approach their study questions. So we just heard some very specific maybe study designs for specific diseases or disease domains, whereas this crowd might be more of a PHIWAS crowd. And there's a cultural divide, I think, in that kind of more, I think, is viewed as a data mining approach. And so there's a reluctance for these resources to be used that way. And that's in contrast with something newer, such as the UK Biobank, which has been very, very amenable to those kinds of really large-scale analyses that I think a lot of us are interested in. So I think there tends to be certain trenches within these more epidemiologic-based studies with the environmental variables that we still want to study.
The reluctance about sharing the data, a reluctance to share the data with people like us in this room, or is it something else?
I think it's I think certain investigators tend to claim certain domains, and it's off limits to any analysis that would touch that domain, that kind of culture. I think some consortia have really worked through those problems. So I was involved in the PAGE study, which was NHGRI funded, and that we did come to consensus that yes, we're going to do PHIWAS, but it took a while for us to work on that. And I think other groups are a bit more forefront about thinking about that. So the Emerge network, they were totally on board with it, of course. So there are just some cultural differences in how people think about hypothesis-driven questions and the use of the data versus more data mining or data exploring approaches.
Carolyn Hutter, NHGRI. As a proudly trained epidemiologist, I'm going to also defend that there sometimes is, and this is advice to this culture, a sort of lack of appreciation of what epidemiologists have been trained to do and what they can bring to studies, which causes barriers to be there. And so come to it with a recognition that that is a field that has expertise that is not always come to with that appreciation by other groups, or that's their perception.
Can I?
Great.
Yeah. Thank you, Carolyn. Speaking on behalf of most epidemiologists, I think that is part of the piece that you're talking about is not so much an entrenchment, but a real difference in what causal inference means and what the evidence base needs to be to move forward. It just is a different culture in terms of the way that we've all grown up in the genetics world and case control kinds of designs and things like that versus cohorts. And they have come together now. It's sort of an arbitrary divide, but it was a real one for a while. The other thing I would say is, as we move from gene discovery to all of the sort of functional annotation stuff that's been talked about today, these cohorts, for the most part, have blood that has been stored for a very long time. And there's a real sort of negativity amongst folks like in this room that, oh, that's not relevant to what I really want to do. That's not going to be useful for my thing. And so, I would say in review, that's what I will hear from epidemiologists say, what's going on with your colleagues? I put this thing in, and they say this will never be relevant, this is never possible. So I think it really is both sides are not communicating where the evidence base is and what things are possible versus not.
I think we really better move to the next thing because it's an important topic. So Katie is going to lead. Can you put that slide up?
I sure can.
Yeah, just move to the next slide.
Yeah.
And then I think, Danny, we're going to want to take notes after this too. So, okay.
Yeah.
Oh, you can't. You haven't been able to see these this whole time? Oh my goodness. All right. Well, we will review them at the end because the whole point of me doing this was so that you could correct me if I said something that you didn't believe in.
Yeah. Okay, so if you wanted to speak and didn't get to, please communicate your idea to us and we'll make sure to get it in or sneak it in under one of these headings. So we wanted to shift a little bit from programmatic silos and highlight a few other silos that the three of us have observed. The first one we've already touched on. In fact, many of these have come up, but I'd like to just focus discussion on them. The siloing of NIH data and the real potential for NHGRI to lead on this. In particular, what is needed to enable centralization of data and integration of data, including customized use or privacy settings. The second one is a technology silo. Era's brought up imaging as an example, but there are other examples of technology silos where we think we come to the problem from a tech point of view, not from a biological question point of view. Oops, we just lost it. And then the last one, which has just been raised but hasn't gotten a lot of play during the meeting, I'd really like to talk about is funding mechanism silos. So the meeting was led off by saying that NHGRI is about half, 50-50 split between large programs and investigator-initiated grants. I don't know the numbers, but our impression that I've heard from a number of people is that there's very few postdoctoral fellowships or K awards, training grants, sort of investment in training. And it seems amongst the investigator-initiated grants and the big projects that there are missed opportunities for coordination and integration. So let's see. Let's actually start with that one because it's gotten the least play, and then we'll come back to technology silos and siloing data if there's any other points on those that haven't come up. So I'd love to hear some thoughts on mechanism silos. First, examples of those or cases where that's been a problem, and then opportunities or suggestions about how to change that. For example, changing the mix of funding, changing the way in which differentially funded projects interact with each other, requirements, carrots or sticks to make that happen. So let's hear some thoughts on funding mechanism silos. Anybody, I'll call on you in a second. Anybody who hasn't spoken yet? Yeah, back there.
Hi, Howard Cheng from Stanford. I want to make a comment about the training mechanism, especially the KOA mechanism for clinical investigators. My understanding is that I think that mechanism is not supported at NHGRI. There are physician scientists and others who are interested in obviously working in the NHGRI space, but they can't have a career path that way because they have to their project would have to then go to a different IC, and then they don't quite fit because their focus is really on genome mechanisms or genome technologies. So this seems to be a gap that hopefully can be addressed.
Great. Across the room.
Hi, this is Tina Gatlin with NHGRI. So the KO8 mechanism, so NHGRI, we recently signed on to that actually a few years ago. So we have funded a few KO8, and the focus of that is genomic medicine. And then we have a similar KO1, so the focus of that is more on genomic sciences.
Okay. Do people think we need more of those? Is it sufficient? Can you hit your button?
It's a small program compared to some of the other institutes.
Okay.
But
so we have it, but
institute.
we need more? Differing views? Or
We're happy to receive more applications. That's more of the issue, is not getting enough fellowship applications, K applications. We certainly have gotten many K99 applications, but that has historically gone up with NIH since the K99 mechanism came online in 2007. So NHGRI has definitely funded a lot more K99s over the last several years.
But Eric is saying there's no cap on these, or Tina was saying. Yeah.
Correct.
Over here, I just want to ask.
Yes, back to you. Thank you.
Just a clarification. When I'm trying to understand exactly what
Each
you
time
mean.
say your name.
Eric Jarvis.
Yeah.
Yeah. Each time, a bunch of Erics here, by the way.
Yeah.
All right. So, when you say silos, I'm wondering if you could give a better definition of what you mean by that, in a sense, because one could think of it, things where there's absence or where there's a bunch of people working alone by themselves and not impacting the rest of the world. So.
Yeah, go ahead.
So we talked to NHGRI about this, and I think we shouldn't beat ourselves up too much. There is a lot of interaction, so it's not that everything is siloed. What they were saying is that the programs are formally disconnected, and when they get together, it's often by accident, whereas or somebody happens to know, or somebody is in more than one program, or they're in the same institution. And so I think they just want to try to figure out how to increase those. And whether, and we didn't really put slides of this, but what mechanism should they do to increase those? So some of those are, an RFA where it's cross institutes or cross projects that formalizes it, as opposed to just making it random or non-random collisions, I guess.
Yeah, to elaborate, an honest opinion that I hear often is that some of the big projects from NHGRI are not worth the amount of money that's invested in them. NHGRI should just give more money to individual labs with great ideas, okay? You may agree or disagree with that, but that's the kind of attitude I'd like to hear like earthed up here and discussed a little bit.
So in this case, the big program project would be a silo. Is that?
Yeah, so the silo would be the mental construct that people have that you are either an investigator-led grant or your Encode project, and those two things are completely different, and they're not trying to achieve the same goal, and they're not talking to each other. So it's a mindset, I guess. That's the silo. Yeah.
So let me comment on that, and I'm part of Encode, and I have been for a long time. So so that was something that people talked about from day one, even when it was in the pilot stage 15 years ago. And NHGRI has pushed the groups, and the groups have pushed themselves to try to end that problem. And I think it's done pretty well because, for one thing, the data are used by thousands of people. We have it's out there for people to use. But also they welcome groups to come in, even if they're not funded by it, to be part of it. And I don't know how many are, but a few are at least, a handful. And that may be one of them. Maybe that needs to be emphasized more. And some of those groups actually come to the meetings and participate in them, in phone calls and things like that I think helps that.
So one thing, knowing that better, then one thing I think that NHGRI could consider that has worked in consortiums in which I have helped lead on, whether it be genomics or neuroscience, is to have an open door policy on big projects. It requires a lot of management to do that, but anybody who's sane, who's not going to be totally disruptive to the group or something like that, be allowed to join the project, whether that's joining intellectually, joining with actually contributing data, or want to listen in. And I'm not sure if that's part of the philosophy, but that prevents the silo kind of feeling, I think.
Yeah, so for, this is Elise Feingold from NHGRI on the Encode project. As Rick said from the beginning, we have tried to run the Encode as an open consortium where we've invited people to join as long as they have funding to do the work and they specify what they're going to do and they actually actively participate and abide by data sharing policies and just being good collaborators. We've always had this since the beginning, and we've had a handful each round. And I believe other projects have done the same. I don't think all of the NHGRI projects have done that, but I do believe it's been replicated in at least several.
So people may not be aware of that, and I think maybe NHGRI needs to make that a bigger deal out of that, so that many more people are aware of it. So.
Not to hog up the time, but I would argue that's not open door enough. That is, if you can only open door if you have money, as opposed to because I think that's what I just heard. If you had the necessary funding to what you want.
Right. If you're going to be contributing in a substantial way, you have to show that you can contribute in a substantial way. If you've got funding for analyses, you've got funding for data generation, it's got to be a two-way street because there is intellectual sharing of unpublished data. Yeah. We don't want people just coming in and not participating fully. So that's what it means to be part of.
And I'm arguing that method we've not used that method. We've had like for the vertebrate genomes project, it really is an open door policy, as long as the person, even if the person just wants to listen in conversations or contribute intellectually.
Okay, noted. We should consider that. Yeah. All right, Gil, wait just a minute. Somebody over here, and then you. I can't see.
Dave.
Oh, David.
Dave Valley Hopkins. To put additional pressure on this, those of us who have training grants in genetics from NIGMS know that they have recently said they will only fund one grant at each university that deals with a specific topic. So, for example, genetics. They recognize genetics as a topic, but they will only fund one training grant regarding genetics at one university. So there's going to be a larger demand for ways to fund individual trainees and programs for trainees in genetics and genomics.
Great, thanks. And
Is that an NIH-wide?
NIGMS.
NIGMS.
Yeah.
Okay.
Gail?
So first I want to quickly comment on the previous conversation. Just, from a data user point of view, data does not want to be confederated. Data needs to be liberated. You want to put your data as much, give yourself 6 months, 12 months, write your first Nature paper. It's genomic data. We can reanalyze it 1 million times and there'll still be amazing things in it. You want to liberate your data, put it out there for people to do whatever they want with it, and you'll get the coolest results out of that. So that's a general comment. I want to make a more specific one, which I hope fits more with mechanisms. Even when you liberate data, you need to think of how you do it, and you need to notice when tools come up that allow you to do it in a better way. So I'll give an example that I think was mentioned yesterday, but I'll elaborate on it more. In Mendelian genomics, right, we have Mendelian diseases make for like 3 to 7% of the general population, right? So in aggregate, it's a huge, each one is rare individually. In aggregate, it's huge. We're going to sequence millions of Mendelian patients over the next few years. So we need to write algorithms that can diagnose our patients. Give me a bunch of reads, give me the medical record, and I'll tell you what the patient has, and the clinician can verify that or not verify that. You can go to dbGaP. There are hundreds of thousands of Mendelian patient genomes there that have been sequenced and paid for by NIH and by taxpayers, except those genomes come with no phenotypes. You get the 99.9% mass of data, and you do not get the phenotypes. You can sometimes get a label that says, this patient has disease X. That's the post-diagnostic label. That's useless, The variants themselves are 100% penetrant. So if you tell me it's a Widerman-Steiner variant, sure, I can find it in there, but your patient then doesn't worth more than 1,000 genome person that I stuck a variant in them. So we don't have pre-diagnostic phenotypes. And then people come in to us and say, oh, dude, that's all PHI data. It's complicated. We can't give that to you. We don't have people to sit down and scrub it. So we build a computational tool. It's always the answers are always in the tool. We build a computational tool. You drop in your genetics notes at one end. It's PHI. You get a bunch of HPO codes at the other end. That's not PHI. But nobody cares. A tree falls in the woods, and everybody's busy, and it doesn't make a sound. So we still have hundreds of thousands of genomes in dbGaP of no value at all. It's shocking. So I wish the funding mechanism would say, dude, you have to have your pre-diagnostic phenotypes in there.
I'm going to call on Molly, who we haven't heard from yet. Thanks, Gail.
Molly Gasparini, University of Washington. Just since we brought up training, I wanted to bring up something I've heard from folks watching remotely and at the trainee level. People seem very concerned about the lack of discussion about the reward systems for trainees and what will certainly become large-scale consortia projects over the next decade, and also specifically how to support maybe a unified math and bioinformatics training and curriculum for trainees across institutions, so the work that we are doing in the next 10 years can be enacted correctly.
Excellent question. I want to hear answers to that from people. Any thoughts? Yeah, Mark.
One thing
Identify
I'll just
yourself.
say, I'm sorry, can you hear me yet? So one thing I'll say
Mark
in
Kirstein.
relation to, is it working?
Yes.
Oh,
Go ahead, Mark.
Yeah, sorry. I just one thing I was going to say in terms of incentives for people and bioinformatics training. I think one thing that people should appreciate is, as we're generating more and more data and it's becoming easier and easier to do that, it's not necessarily becoming easier and easier to do analysis. And I think a lot of times, people often feel, who do a lot of analysis work, unappreciated for the amount of work that they have to do and also the sophistication of the work. And I do think that we have to see value in data, but also value in analysis. And I don't know how to quantify that or get to that, but I do think that's a very important and a very data-oriented culture, thinking about that.
Okay. Are we done?
Yeah.
Okay.
We have to end.
So great discussion. We need to end to move on. I think Elise is going to lead the next session, and maybe some of the other ideas can come out in that. Thanks, everybody, very much. That was really useful.
I'm not sure where to save it on this.
Yes.